are waiting to get live, and we are live. Hello, everyone. Good morning. Again, uh, this video is again going to be a little digression from the kind of videos that I make on business learnings. In this video, I'll talk about my little exposure to research and science that I had during my undergraduation at IIT Guwahati. Uh, well, I studied biotechnology in my undergraduation, and as you know, in 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 your fourth year, you have to do a research project based on your what you like, what you want to work with, and the kind of projects that might interest you. So I went through the profiles of all the professors in our department, and I narrowed down to you know one prof, Dr. Vikas Dubey. He was working on Kala-azar, the disease Kala-azar. If you're not aware of it, so it's a lethal disease, a severe disease, and the problem with the disease is, uh, about 500,000 people worldwide are at the constant risk of getting Kala-azar, and in India itself, hundreds of people die every year because of this disease. It is not reported because the people uh, who die, who suffer the most, are come from underprivileged background, uh, from Bihar, from Africa. Most of the cases are from Africa, so there's not much incentive, not much funding in the research. That is a core problem. Uh, Another thing which makes this disease very difficult to cure is the parasite Leishmania donoani, which is responsible for this disease. It keeps on changing, you know it keeps on adapting to various drugs uh, that scientists develop to cure it, to get rid of it. It develops resistant. Its genes develop resistance, and the medicines do not work. You have to constantly keep changing the medicines. That's what the problem is. uh, Coming back to the research, so our professor was working on finding vaccine targets, drug targets of this disease. What are drug targets? I'll tell you. Um, so this parasite, let me just first explain you how this disease works. So it is carried by sandflies, and when they suck blood out of their prey, us human people, they also inject this parasite into the bloodstreams. And when these parasites get in the bloodstreams, they, you know, I'm simplifying a very complicated concept. These parasites keep replicating inside the body of us humans. Especially, they invade our immune cells. Immune cells are the ones which keep us healthy. Cells of the immune system, which help us fight against all kinds of diseases. This parasite attacks those cells, kills them, and as you're familiar with AIDS, what happens with AIDS, HIV/AIDS, uh, similar thing happens. The HIV also, kills the immune cells. There is a bit of difference between these two diseases, and the person finally dies with some other disease because his immune system is not very good. And this is what happens with Leishmania donoani. The person usually dies with another disease, opportunistic um, microorganisms which are there to invade your body when your immune system is not very strong. So this disease works in that way. Imagine the kind of horror. You're not able to defend your body. So our professor was working on searching on various vaccines, various chemicals that would, prevent this Kala-azar, that would prevent this parasite to replicate, to do functional activities in our bodies. And if that was his work, identify novel drug targets in in the parasite. So These parasites are made up of proteins. They connect to each other with proteins. They need proteins basically for everything, just like we need proteins for everything. And please do not confuse these proteins with the whey proteins. They are little different. Uh, so for functionalities, there are different kinds of proteins, thousands and millions kinds of proteins that a bacteria can have for its functionalities. Everything. Uh, for every functionality, there is a protein. One protein will help you replicate your DNA. Another protein will help you break things apart. Another protein will help you divide. And there are numerous proteins for numerous kinds of works, and these proteins keep on updating. Okay, so these proteins, how are they made? They are made from translating your DNA sequence. So a bacteria also has DNA, and all those proteins are made up of reading those DNA and then aligning the amino acids based on the patterns found in that DNA. And how does a protein work? So protein is basically a sequence, like you have seen. A DNA is a sequence of those peptides. Right? In the similar way, a protein is a sequence of amino acids. So there is interaction between different kinds of amino acids, and based on these chemical interactions, these proteins fold in very distinguished way. Every protein has a distinguished structure in which they fold, and this structure helps them do things like the structure of my hand. This helps me write if I do it this way. Similarly, there are proteins which fold in such a way that they help they help getting some things accomplished, right? Now, uh, okay, so what we want to do, we want to target these proteins so that the bacteria, sorry, the protozoa in this case, protozoa is a kind of microorganism. The protozoa is not able to perform its daily functions. It's not able to replicate. We want to target those proteins which do not allow this protozoa to replicate, or do not allow it to do anything else, a lot of things, so that it cannot, you know even if it enters a human body, it is not able to do anything else. It's not able to kill those immune cells anymore. That is our target. And we want to find drugs as in chemicals which can do such a thing, which can make a protein useless. Okay, so protein has this shape. Let's say a chemical comes, binds to it, and breaks its shape, somehow twists, turns, breaks, does anything, so that its basic structure changes, and it is no longer able to uh, perform what is it is supposed to perform. And therefore the bacteria, uh, or the microorganism in question here, the parasite, uh, it simply cannot perform its daily function, so the human is saved. The problem is the ones which get killed are not replicated, and the ones which are alive at the time of DNA replication, at the time of reproduction of those parasites, there are mistakes in copying the DNA, which causes mutations to occur, and which causes the proteins to change. And these changed proteins, our drugs cannot identify these changed proteins anymore, and the parasite is now, immune. Our drugs cannot affect them, so they go ahead, destroy the immune cells, and this is what happens. So we constantly have to develop new drug targets. So this was the entire work, development of new drug targets, and to do that, we have to constantly identify the proteins within that protozoa. I hope all the points are clear up to this point. So uh, a protein has a sequence, okay, and scientists have listed down all the proteins that a microorganism can have, especially for Leishmania donovani. Back in 2014, we had a list of all those proteins which it had. Okay. Now we uh, list of proteins as in you know it has this DNA, and this DNA gets translated to this protein sequence, and I have the list of all those possible protein sequences. Okay, these are possible proteins which a bacteria can have or has. The problem is we just know their sequences, we do not know their structure. And since a protein is characterized by structure, it functions by structure, it's difficult uh, to just predict how a protein will function just by knowing its structure. There are various tools available, we'll come to that. So now my job was to figure out uh, what are functions of these proteins. Okay, so back in 2015, we had about 5,000 proteins, uh, and we wanted to calculate the functions of all of them. There are various steps to calculate the function to arrive at to best estimate the function of a particular protein. Uh, so there could be ways based on the sequence, based on the sequence that is present at the head, at the initial, based on the sequence that is present in the middle, based on some patterns which are existent throughout uh, the chain of the protein. Based on these things, uh, scientists have able to find a correlation with presence of certain patterns, sequences with functionalities. They are not 100% sure. Of course, with some confidence, they are able to predict what this protein might do. Now, there are various tools for it. Various universities, various professors and scientists have been developing that, and there are 50s of tools. Back in my time, there was a tool. There were about six, seven, eight, 10 tools to predict the physical properties of the protein, some 10 odd tools to predict the chemical properties, some 10 odd tools to predict the structure of the protein. So similarly, there were a lot of functions and a lot of for every function we had a lot of tools, online tools, which will tell you about the properties. What I did, I'll tell you what I did. So I listed down all these tools that we had. Parameter one, property one of a protein, and this property could be um, this particular property could be, you know, uh, this particular property of the protein. This could be determined by these many tools. Property number two could be determined by these many tools. I individually went to all these tools. All you have to do is get the protein sequence, input it, and get the results. Okay. There are algorithms working. We do not know all of them. We do not know how accurate every one of them is. And based on those results, I'll know what are these properties. So for every protein, we get about 50, 60 properties, uh, 5, 6 properties, every property from 10 sources. We will take the consensus, and at the final level, based on different kinds of properties, we would like to estimate what the function of the protein might be. So this is a tedious process, a long process. I started working on it back in September, and my placements were also approaching. A lot of things were happening back in that time. I was not able to devote as much time. I I predicted the functions of about 10 odd proteins. Out of 5,000, I was able to predict only 10 proteins, and I had decided that I would like to integrate this in my next, that is my eighth semester. I tried it. Uh, well, what happened in my eighth semester? We have 50 sources, and what I'm proposing is a software which integrates the tools from all these 50 sources, gets the protein, queries across all these sources, and gives you an output which combines, takes the consensus, and gives a final output. So if it were done at that time, it would have been a major breakthrough, I would say, because a lot of scientists, professors were constantly doing this work manually, and it took anywhere from Anyway, it took several hours to get it done. If you do it, you have to go through 50, 60 websites, and each has a different format. Each will give output in a different format. The results will be in different formats. Some will be in paragraphs, some will be in standard, some will follow a standard, some will have API, some will not have API, some will give you the option for the JSON output, some will give you in XML. Lot of inconsistencies across all those formats, all those websites outputs, and that posed a lot of difficulty. I even started working on it. I integrated several of them. A lot of challenges happened. I could not finish it. Unfortunately for me, I could not finish the project. And at the later part, I started working on it manually. I know it was impossible. Uh, I had done, I had predicted functions of 10 proteins in my previous semester, and that had took me several days, several full days to work on. I extracted several weeks from it, from a timeline. I think about 50. I devoted about 15 full days for this work, and I predicted the function of 64 proteins manually. It was a tedious job. It was a very repetitive work, mind-numbing work. But I was sure that if I go ahead and code, it would take me several months, and I did not had that time. Unfortunately, with all the courses going on, uh, that was I was not able to continue on this project, and I had thought about returning to it, but I never did. So that was all about my contribution. Predicting functions of of proteins of this parasite, Leishmania donovani, which which is notorious for killing thousands of people every year throughout the world. So this was all about me, my BTP, BTech thesis project. A lot of learning were there. You can say it was kind of failure, but I was able to accomplish something. Thanks for watching if you have been watching so far. Bye.